And good day, ladies and gentlemen. I'm Hazima Mozam, and I welcome you to Comstech's Distinguished Scholars webinar series. We are very delighted and honored to have Professor Dr. Samar Hassnain with us. Professor Dr. Samar Hassnain is a leading structural biologist with over 45 years of synchrotron radiation experience. The title of his talk today is Advanced Methods for Structural Determination of Proteins and Their Complexes. Drug discovery for motor neuron disease to COVID-19. With this, I would request His Excellency, Professor Dr. Iqbal Chaudhry, to kindly, for his kind welcome address.
Bismillah ar-Rahman Rahim. Good morning and Assalamu alaikum to everyone. By all definition of being a distinguished scholar, we are absolutely delighted that we have such a wonderful personality delivering a lecture today. This series of Comstech has attracted lots of attention, and we have been very fortunate to have some of the most distinguished scholars delivering lectures on most exciting topics of science and technology today. But for introducing such personalities with tremendous contribution to the field and their stature in the field, it is more appropriate to select someone of the same stature who come forward, volunteer, and introduce his or her peer. And I'm absolutely delighted to request Professor Dr. Christian Berzel, a distinguished scholar from University of Hamburg, Germany, who worked in the field of structural biology and has lots of very good work to his credit. Some of the most fundamental discoveries made in the field of structural biology came from his laboratory. And he is the most appropriate person to introduce Professor Dr. Samar Hassnain, today's speaker. So I would request Professor Berzel for the introduction of Professor Dr. Samar Hassnain.
Yeah, dear Professor Chaudhry and best friend, dear organizers, colleagues and friends and all participants of today's Comstech Distinguished Scholar webinar, it is my particular pleasure to introduce today's speaker, distinguished Professor Samar Hassnain. We know each other for many years, and I just summarize his CV, and please correct me later if something is missing or wrong. Professor Hassnain is performing research for more than 40 years in structural biology, particularly utilizing synchrotron radiation and associated techniques applied in structural biology. As key techniques, I would like to mention X-ray crystallography, X-ray solution scattering. And I remember where he is an advocate of XRFs, I come back to this, as he analyzed a number of metal proteins in detail. And today, of course, he's applying also cryo-electron microscopy as complementary technique. He can certainly be assigned as a pioneer in the use of synchrotron radiation for and in biophysics, in particular structural biology. And in this context, he also pioneered and contributed to synchrotron radiation facilities and beamline instrumentation. So he obtained his PhD in experimental physics at the University of Manchester, already working and applying crystals and exposing crystals with X-rays. He joined as fellow and postdoc DESY. DESY is the abbreviation for Deutsches Elektronensynchrotron. I'm today on the DESY campus. And at this time, it was one of the first synchrotron facilities, today called, for this reason, first generation facility. The main use at this time was for high energy physics and corresponding experiments, however, producing already very useful radiation to look at biomolecules. And just to comment, along the last 50 years, DESY, as many other facilities worldwide, they changed from high energy physics to photon science. And with the experience Samar Hassnain collected at DESY, he joined the UK efforts to construct one of the first dedicated synchrotron facilities close to Manchester, the Daresbury National Laboratory. I visited myself as PhD student and later on scientist several times to collect data. And he headed the biophysics group at the Daresbury lab. And this is then called, or was called, the second generation synchrotron radiation source. It was running for 28 years, very productive up to 2008. And I remember we met in Hamburg at DAISY in the late 1980s of the last century, we say today, in 1990s. I was at the EMBL outstation, the European Molecular Biology Lab, first as beamline scientist and later group leader using the storage ring DORIS 2 at this time. And Samar Hassnain frequently visited DAISY as radiation user, experiment advisor, and I remember also reviewer in terms of the EMBL annual reports. And we met several times. This time was somehow most groundbreaking for me and maybe I guess for our age group, as the first efficient electron error detectors came up for data collection and processing. Before X-ray film was in use, and data collection at this time was a few days, sometimes painful, long, and you need chemicals and all this stuff. And after you had only film. Today we collect very efficient diffraction data at synchrotron radiation in the range of seconds. So this is a tremendous change. And the image plate detectors developed at this time in the late 90s by Jules Hendricks were pioneering. I remember his name because the image plate detectors are till now in use. They're very robust. And Jules Hendricks, later founder of MAR Research, unfortunately passed last year by COVID-19, the topic of Samar Hassnain presentation today. After 2008, Professor Hassnain joined the University of Liverpool as Max Perus Professor of Molecular Biophysics, and he established and headed the Barkla X-ray Laboratory of Biophysics. Barkla is well known as physicist. He investigated and characterized X-rays and called Röntgen radiation and obtained the Nobel Prize in 1970. And to complement his CV of Professor Hassnain, he initiated in 1993 the well-known UCR journal, International Union for Crystallography, IUCR, of synchrotron radiation. And in terms of this honor and duties, he is editor-in-chief of a number of IUCR journals. And coming back to this, as I remember well, one of his early most applied techniques was EXAFS. EXAFS is abbreviation for Extended X-ray Absorption Fine Structure, a most useful technique and method to analyze particular structures of around metal ions in proteins to high resolution. And because one of his main interests he's involved is biological electron transfer, the nitrogen cycles, and neurodegenerative diseases. And today he's involved in a number of structural biology and structure-based drug discovery investigations targeting neurodegenerative diseases, malaria, and also COVID-19. He has a number of very high impact factor publications in 21 on this topic. And this brings me and the audience back to the title of the presentation mentioned before, Advanced Methods for Structure Determination of Proteins and Their Complexes Applying or Drug Discovery from Motor Neuron Disease to COVID-19. With this, I hand back or over to the chairman and chairwoman.
I assume I should start. So,
please.
Let me first of all thank the organizer for inviting me and giving me the opportunity to join in this prestigious webinar series. I'd like to also thank Christian for a wonderful introduction, as well as reminding me of many good times which we have had both at Daresbury and in Hamburg and elsewhere. So let me just try to put the scenario for today's talk. I'm going to divide the talk into 3 sections. The first section is going to be all about the advanced structural biology methods for a determination of proteins and their complexes. I'm going to focus on 2 techniques. One is the X-ray crystallography, and the other one is going to be Cryo-Em. And then the second part, I would divide that into 2 parts. The first one will be on superoxide dismutase and its relevance to motor neuron disease, and the work which we have been carrying out for more than 30 years or so, what that has led to. And then the last part is going to be about COVID-19, where I'm going to focus on main protease, and this is an example of rapid response in terms of the research, because this is something which we have never thought about doing until 2020, and then what we've done in the last 12 months or so. It is obviously, I'm going to try to give credit to a large number of people, but the protease and COVID-19 is a very, very rapidly changing field, and huge contribution from structural biologists. So on this panel, you see the picture of the superoxide dismutase on here on the top, and on the left hand side you see the main protease, which is a dimer, and is shown, one particular monomer is shown in a single color, with domain 1 and 2 forming the catalytic pocket, where his cysteine and histidine is highlighted. So as Christian introduced, we established Barclay Lab in 2011, which is 10 years ago, and we make extensive use of diamond, Soleil, Springate, and X-ray free electron laser, which is co-located with Springate. On the left hand side, I'm showing you a Cryo-Em. This is one at Leeds, which we make extensive use, and then we also use the Diamond EPIC facility. So let me see whether I can move my slide to the next one. So we are fortunate as a structural biologist that we have a number of techniques which are available. In the context of today, I'm going to briefly mention the third technique, which is the NMR, for which HEJ and Karachi has a fantastic set of facility. X-ray diffraction or crystallography requires crystals, and we can use X-rays from sources such as Barclay Laboratory or a synchrotron or an X-ray free electron laser, and we obtain lots of diffraction images which we then can bring together to analyze through Fourier analysis to determine the structure. NMR, in contrast, looks at solution, but it is restricted to relatively smaller proteins of less than 30 kilodalton. Cryo-electron microscopy is applicable to relatively larger protein. It becomes a struggle at the moment if the size of the protein is less than 90 kilodalton. It is particularly powerful for membrane proteins and for much larger complexes. And I'm going to try talking to you about that as part of introduction. Just to give you a perspective of where we are in structure determination, in this particular graph, I'm showing you in blue, starting from 1999 till the August 2021, the number of structures which have been determined by X-rays, NMR, and then in gray we see the number of structures which are coming through from cryo-electron microscopy. On the top, just for ease, I've just shown you 2 time points, 2010 and 2020, for the 3 techniques. And what you see is that in the Cryo-Em case, the growth is 50-fold in a decade. And this is one of the reasons in 2015 I called Cryo-Em revolution the method of the decade, and wrote it in my editorial in 2016, bringing Cryo-Em within the IUCR-J, which is one of the latest and one of the premier journals of IUCR. NMR has continued to make very important contribution, but the number of structures which are determined has remained relatively stable over the last decade. Despite Cryo-Em emergence, the number of structures with X-rays continue to increase year by year. As you can see, in the last 5 years, the structures have remained relatively stable, but increasing significantly in 2020, and that seems to be the case in 2021 as well. So let me just give you a bit of an overview of what you need. I've done this for students who may not appreciate all the steps which are required. So there are 3 key steps which is required in X-ray crystallography. The first of these is common to NMR and Cryo-Em, that is to be able to express a protein and then get a significant amount of pure protein. And for this chromatographic, there has been significant improvement in chromatographic methods, so much larger and higher quality of protein can be obtained. Of course, molecular biology has transformed the ease with which we can express and clone proteins. The second step is a unpredictable step in crystallography, and that is to be able to grow crystals and then harvest them to expose it to X-rays, which could be which could be in synchrotron or X-rays. And then once you've collected large number of images, then you put them together through Fourier transform into atomic models. There are lots of other steps which are involved. I don't have time to talk about it, but this is the basic aspects. So a step of producing recombinant proteins, some of you would have already got experience for it, and this is an example of a wonderful metalloprotein copper nitride reductase, where you can begin to see already that the E. coli cells look healthy and the colors are already beginning to appear. When you go through the chromatographic steps, you can see the blue column of the protein here, and when you purify, it's a pleasure to work on such proteins. It is important to understand your protein well before you begin to start crystallizing, and that is quite important. I don't have time to talk about the incorrect, if you don't take care of your samples and good quality, then you may end up looking at protein crystals which may not represent the protein you expect to represent. There are various methods of crystallization: the hanging drop method, the sitting drop method, and dialysis. And you can grow different size and shapes and quality of crystal. This is an example of copper nitride reductase, which have been grown for several years in our lab. But we have to adjust those crystallization to suit the method we're going to use. So the one on the left here I'm showing you, which had to be optimized in this particular shape to use a technique called SF-ROX, which uses the XFEL, but it uses the rotational crystallography, and where you walk through the crystals and get lots of images from one crystal. XFEL is so powerful that you get only one image per shot. And that, of course, is a huge advantage in terms of providing you damage-free structures. So, whereas these small crystals here may be still too large for serial femtosecond crystallography, so you've got to work on it to get smaller crystals. Whereas these crystals, which may not look very beautifully formed, but if you can price these crystals, they give you atomic resolution in our case. So again, the three steps: you have crystals, diffraction, electron density, and atomic models, and that is the accuracy of atomic models is as good as the data you collect, and that is predetermined by the crystal quality which you may have in combination with the sources. So let me just give you some examples of structures by X-ray crystallography. On the right-hand panel, I've shown you the they are on a scale. So this is at the bottom is 200 angstrom is labeled. So there are some protein structures which you would be very familiar with. And one of the first protein structures was myoglobin, which is shown on the left-hand side, second from the bottom. So that's here, and then hemoglobin is on top. So now you can see that even though they won the Nobel Prize in 1964, we have come a long way, and we can determine very complex structures and much larger structures with ease to a very high resolution. One of the first GPSR structure, rhodopsin, was determined in 1999. What also I show you here on the left-hand side that you can do by determining a structure, begin to understand the function of the molecules, and then therefore changing the understanding in terms of biology. So the face of biology has been transformed through structural biology over the last 30 to 40 years. We think of biology very differently once we know the atomic shapes and the atomic and the underpinning structures which define the chemistry or the mechanism of the system. The structure also provide you an opportunity for doing knowledge-based drug discovery. And this is an example I'm giving you at the bottom, that the structure of main protease of the SARS-CoV-2, the COVID-19, was determined within 2 weeks of the release of the genomic sequence of the virus. And that's really is amazing how quickly the world has come together to do this. And the data was deposited, made available prior to publication. This particular preprint received much higher citation already before publication than many many papers which are of high impact which they receive. Large number of structures have been reviewed recently in IUCRJ, and since then, as many more structures have emerged for this particular protein and under different arrangements and through large high-throughput screening. Our chairman, Christian Bessel, has made tremendous contribution in COVID-19 main protease and for the search for repurposing of the drugs or the fragments. They also have very strong links with looking at the natural compounds which are provided by Professor Iqbal Chowdhury and his laboratory. So let me just give you a perspective of the impact of synchrotrons on crystallography and biology. In this context, Sesame, which is Pakistan, is a founding member, is critical for several of the comstech members. We have more than 150,000 structures of macromolecules and complexes, several blockbuster drugs. The other measure which you have is that several recent Nobel Prizes, 6 are listed here, for chemistry and physiology have been obtained using synchrotron radiation. So it's a very powerful approach to use. So let me just briefly then move on to give you an overview of protein structure determination using Cryo-Em. So the first step is the same, which is to be able to express and purify a protein. In this case, I'm showing you a membrane protein, which you then take, instead of forming crystals, you spray them on a grid and quickly freeze. And so this is frozen solution is what you're looking at. And then you look at the single particle imaging and then use many of those images to combine, to produce a 2D projection and a 3D model, and if you are lucky, to get high resolution structure. And in this context, generally when we mean high resolution structure, we're talking about 3 angstrom resolution. But the 3 angstrom resolution, the map quality in Cryo-Em is significantly better than the 3 angstrom X-ray map. Generally, the 3 angstrom Cryo-Em map resembles more like 2.5 angstrom X-ray map. So these are some of the examples of the Cryo-Em structures which is given. So you can see very complex structures can be done for membrane proteins and their complexes. So this is an example on the top I'm giving you is for the tryptophan channels. Several structures have been done over the last 5 to 7 years, starting in 2013. Rod McKinnon himself has been involved in many of these structure determinations. Many of these are reviewed in Science in this particular review. So these are already very complex structures, and because you're not required to make crystals from them, is significantly easier to achieve these large complexes. One of the largest complex, which is shown on the left-hand side, is the mammalian respiratory complex 1, 3, and 5, and you can see the molecular arrangement at reasonably high resolution. This currently will not be practical or feasible, even with decade of effort to achieve something similar. This is a real unique aspect of Cryo-Em, where you can begin to look at the whole of respiratory, for example. We've been involved with this structure of QNOR, for which we and others had determined from different families, including Neisseria meningitis, crystal structures, but all of the crystal structures had shown it to be a monomer. When we obtained the structure, it was very clear, even before the detailed analysis, that it was a dimer indeed. So the sample preparation I've explained already, and what you want to do is to form a very well separated particle arrangement in a vitreous ice layer, which you then subject to get images, and these are single particle images which you bring together, and this is, you can see already from this raw images that the molecule was a dimer indeed, not a monomer. So you didn't need to even get through the final analysis, and then by combining those you can improve your resolution. So let me go from one revolution of Cryo-Em to what the other revolution is taking place, which is in the X-ray field, where X-rays have gone from lab sources, which have improved along with the detectors, but the synchrotrons have gone from first generation to the fourth generation, where the emittance has become very, very high. So the brilliance is extremely high, emittance is very low. And this has kept on driving the community to get to as high a brilliance as possible. So round about the turn of the century, it was argued that if you could produce a laser in the X-ray regime, then it would be that would be the ultimate brilliance we can reach. Now, in doing this, the arrangement is X-ray free electron laser, and there are now several in operation, including where the chairman is residing in Hamburg. So this is a X-ray pulses, which is being shown in Sakura, which is the Japanese X-ray free electron laser. It was the second which came online. And these X-ray lasers, together with pioneers such as John Spence, who was not only had a pioneering spirit, but also a fantastic scientist who was always prepared to help and open whatever he discovered as quickly as possible to others. And it is he, along with his colleagues, is very largely responsible for opening up serial femtosecond X-ray crystallography, which is now extensively used. Unfortunately, John passed away this year, and if you want to know more about it, there is an obituary for him in IUCRJ. In contrast to synchrotron, each pulse of light which you receive is different in the X-ray free electron laser. It is very much like synchrotron, pulse synchrons, which was the first generation sources, not the storage ring, which is the norm. But you can see the pulse per energy is 400 microjoule per pulse, which means that once you put one X-rays through your crystal, it's not going to be there. So this is just showing you here in a normal light source, you have each pulse has a different phase. So, but the pulse brilliance have been increasing as we've gone through the last 4 decades or so. In the X-ray lasers, all of them are in harmony and then giving you coherence, which you can exploit for doing diffractive imaging. Unfortunately, the promise of imaging macromolecules without crystallization has not yet materialized to high resolution from X-ray lasers because the intensity is still not sufficient. This is the Sesame site I'm showing you. For those who have not been yet visited, it is in full operation even during COVID time. Next call is due to come, so do apply and get access to it. And this is spring 8, the synchrotron, which is the circular, where the same set of electrons keep going round and round for more than 24 hours or so, whereas in Sakura, there is a repetition rate of 30 hertz or so. And X-Files have provided another 10 billion times in peak intensity or peak brilliance compared to the best synchrotrons. So the idea behind SFX, which was promoted by John Spence, is that you capture the image before the sample is destroyed, And that can happen because the X-ray pulses are very, very short in duration. So they are 10 to the minus 15 seconds, that is 10 to the 10 and 20 femtoseconds. So these are some of the references which I've given here, which I suggest you can have a look at some of the examples. There are some great examples which are given here at the bottom, this particular one I've given is for the photosystem 2, where people have been able to use a pump and a probe laser. So pump is a visible light lasers, and then the probe is the X-FEL laser. And by changing the delay, you can monitor the whole of the Cox cycle from going from S0 state to S4 state. There are also mix and inject approaches for looking at substrate binding or looking at turnover, and where the reactant and the sample are use different capillaries, and they mix together in the mixing chamber, and then they are ejected out. So these are different types of sample arrangements and delivery arrangements which are given. This was largely pioneered by John Spence and his colleagues. The hybrid methods are being looked at by many, whereas the rotational crystallography has been mainly championed by the Japanese Sakura source, which we've been using more extensively. So the example of SF rocks is such that the crystal, as I was showing you before, you walk across it for each image. So each image gives you, each pulse gives you only one diffraction image. These are still images which you combine. And you, at the bottom on the right hand side, you can see the holes which have been drilled by the X-ray free electron laser pulses. And this provides for metalloproteins unique opportunity for obtaining damage-free structures in many of the catalytic states of an enzyme. So there is no other method which can provide you damage-free structures. So I'm not going to talk about a structure-function relationship, but I'm going to talk about the structure-based drug design. So this is a cytochrome BC1 complex, which is a validated drug target for antimalarials, and this is an example where you have a biological space, which is your target protein, and then you can screen the chemical space through variety of means by having drug candidates or fragments, and then increase the chemical space in terms of doing that. Now, on the right hand side, one example I give you here, this had to be pulled out in the final clinical trial because of the toxicity. The drug was developed on the assumption that it binds quinol binding site, but there are two quinol binding site, and it was assumed that it would bind quinol 0 site, and X-ray showed, structure showed that the reason it was toxic, that it doesn't bind to QO site but QI site, which is very similar to the human BC1, and therefore it is toxic, and then it had to be withdrawn. So a knowledge-based direct visualization of the target with the drug candidates is important, and using it to improve it. There are now several blockbuster drugs which have been which are on the market, and in many cases reducing the cost of new drugs. So these are this second slide shows you a number of drugs which have been based on fragment-based discovery including some for skin cancer and some for breast cancer. So in today's talk, I'm going to talk about two examples. One which is has a history of almost close to 20 years on superoxide dismutase, as reflected by some of the key papers just of our own group for motor neuron disease and drug discovery. And on the left hand side, the first one is ours, but all others are from different groups, including our chairman, Gunther et al. In Science, which has appeared just very recently. And the idea behind this is that that inhibitor, you find a molecule or a fragment which begins to stop that disease-causing properties, and that's the aim of the drug discovery. So motor neuron disease affects a lot of people. Don't have much time to talk about in detail, but I'll give you some examples of people. So Steve Hawkins, who was diagnosed in very early age at 20, but he managed to survive for a long period. And there are particular reasons why despite his disability, he was able to contribute significantly to physics and science and to society in general. At the bottom is Justin Yerbury, who had a family history, and after once one of his close relative passed away, he decided to change his interest and began to work on motor neuron disease himself. But he fell victim to the same disease a couple of years ago. We collaborated closely in 2018, publishing this important paper. He is still with us, but he's wheelchair bound and is still running a lab. It affects many athletes. There's some discussion going on that maybe extraneous physical exercise combined with genetic disposition may be also a risk factor. So this is the example of three football and rugby players, which is given here. They're all alive at the moment, but they've all been diagnosed in the last 3 years or so. And they each are working very hard to make MND a major awareness campaign, but also to raise sufficient funds so that the research can continue to be done. Those of you who may be interested in Top Gear, this is a Stig who passed away 5 years ago, and he was also a Daniel Craig's 007, double in Quantum of Solace, won many Grand Prix, but he unfortunately passed away at the age of 52. These footballers and rugby players are all in their mid-30s. So just to briefly say, I'm going to focus on a couple of mutants here. So I'm showing you at the interface alanine 4, which is mutated to valine. The survival time for this is very short from the disease onset, 1 to 2 years. Whereas the copper ligand, where the catalysis takes place, the average survival is 17 years. This is what A4V is one of the most common mutation in the North American population, whereas his 46R is one of the most common in the Chinese and Japanese population, and to some extent the British population. So from years of work, we immediately recognize is that the protective loops of these beta barrel is if they are compromised through mutations or the copper incorporation, then that will play a major role in aggregation, and we need to really arrest that quickly if we can through intervention. And this we were able to locate a repeat in APO superoxide dismutase, which is the same repeat as the alpha-synuclein, giving confidence that this strategy was the right strategy. That is, that you need to try to stop the monomerization of the protein, because otherwise the beta sheets will not remain protected. So key references I've given you here, but I've also given you in the previous slide. One of the recent examples where we've looked at the potential of binding selenium-based compounds and to see whether they rescue. And it seems like that the epsilon binds to cysteine 111, and therefore it destabilizes the molecule, and monomerization of A4V is inhibited. And in order to do this, we employed a number of techniques, including DSC. But in order to get accurate results, we modified one of the cysteine, the cysteine 6, to a serine, so that we could get a good measure of which of the compound was giving us the best delta T shift. And then we use this to begin to look at a number of selenium-based compounds, and this is some of the examples which we have, and this was published last year in eBiomedicine. And we said in this particular paper, in the conclusion, in the summary statement, that in the ability of these compounds to target cysteine 111 in SOD may have wider application for protein molecules where cysteines may be involved in pathogenic aspects or promotion of disease. And we identified main protease as a key target to try. So this is just an example of what the epsilon is doing, and associate compounds, how it is rescuing providing the neuroprotection in the mouse N2A cells. At 1 micromolar, we are restoring the activity of A4V to the same level as the wild type. It obviously becomes a bit toxic at higher dose. So therefore, giving promise that 1 micromolar for a number of these compounds may be a good dosage which we can aim for. And we tested this with transgenic mice, which is a good model system for motor neuron disease, and we found that the disease onset could be increased by 10 days, that is 10% of the lifetime of the 10% of the disease onset, which is a significant increase. But the survival time did not appear to improve. We've recently been trying 2 additional compounds which had better neuroprotection in the neurocells, and these results are encouraging, providing a significant delay in disease onset, and it appears that they are going to be increasing the life expectancy to some extent. So in order to see that target engagement is in the cell as well, what we have done, and this is a clever experiment which Rahila Awais and Kangsa have been doing, where the protein has been linked to a venous flytrap at the C terminal for one monomer and for the N terminal for the other monomer. And when they come together, they flourish. And what we see on the right-hand side here is that epsilon reduces the inclusions, which are less than 5 molecules or greater than 5 significantly. And then on the right-hand side, you can see the other two compounds are behaving even much better, with almost MR626-2 providing very few inclusions, larger inclusions. So this is highly promising, and we're going to take this forward. So let me just, in the final furlong, talk about protease, and I'm not going to have enough time to talk about the biology of it, but all of you know that it doesn't have its own ribosome, so it needs to get to hijack its host ribosome. And the two proteins which have been identified very early on as key targets is the spike protein, which is responsible for the entry, whose structure is determined and which has provided a major input into the vaccine design and vaccine arrangements. And we are fortunate that the vaccines are available and they appear to be working extremely well. And the other target is the main protease, which is important for replication. So when we started, the number of confirmed cases were in tens of millions, and unfortunately now, within a year or so, we have over 220 million infection cases with over 4.5 million deaths, which is phenomenal and is everywhere in the world. In a globally connected world, despite lockdowns, it continues to impact our life. So on the left-hand side, I'm just going to be slightly parochial because I have better data for it. In the UK, you can see there have been several cases, several time points when we have had major increases. So we had the first one is starting in February, and then going on to 2021, and again in January, and then currently what we have experiencing. The vaccines have made a major impact. You can see that by the slide on the right-hand side at the bottom, where the hospitalization and the 7-day average in September 2021 is very, very low. And one of the reasons for the success is that the vaccination is close, well above 80% for the whole population. This is for data above 16 and above. And I think we're rolling out vaccines for 15 years old soon, and then for a significant proportion of 12 plus. So main protease is important. There are two proteases, pepin-like protease and the main protease. Main protease cleaves polyprotein and at 11 positions, whereas the PL protease cleaves it at 3 positions. And so we decided to focus on main protease. And one of the first things we did was to try a number of compounds against both the protease as well as for the viral replication. And we were surprised that they were at that point as good as anything which had been tried at the Wuhan Institute of Virology at that point. And our best compound was 1.7 micromolar against the viral replication. So it was clear that it was acting against the main protease, but also against the virus. So the next thing was to be able to show that we could, the target engagement was there. And in order to do that, several groups had tried to do co-crystallography of epsilon with main protease, but had not reported a success. All of the information was computational. We obviously purified the protein, grew the crystals together with a number of compounds. We were successful in growing crystals with epsilon as well as MR631-2. But the surprise was that the compound was absent, but selenium was present. So we did the anomalous dispersion at the selenium KH to ensure that selenium, it was selenium which we were looking at. We repeated this several times to make sure that the data was hard, and once we were convinced, that we then decided to communicate to the world. We also tried to see whether what we were seeing could also be produced in solution state, and this is looking at the mass spec here on where you can see the selenite standard is giving the same peak as we receive on MPRO incubation with epsilon. And so what we propose is that the hydrolysis takes place such that the selenium is left behind and the rest of the molecule is liberated. And the different efficacy of different compounds results from being ability to deliver the molecule and then bind initially where it cleaves away, making the main protease inhibited significantly. So epsilon is now being taken up with the support of NIH grant by Sound Pharmaceutical for phase 2 clinical trial using what was developed for neurological disease for treating patients with moderate and severe COVID-19. The trial will complete in summer 2022, when we hope that one of our front runners could be taken up further. It just remains me to say thank all of you for your attention and to acknowledge research collaborators on the main protease project, starting from Professor Paul O'Neill, Xiaoling Ming, and then the Chinese team spanning from the Wuhan Institute of Virology to Shanghai Institute for Immunology and Shanghai Tech, and then on motor neuron disease, Paul O'Neill, Rahila Owace, and Watanabe and Yamanaka from Nagoya. Thank you very much indeed.
Yeah, thank you very much for the nice presentation. You covered the structural biology methods from giving a little bit of history up to now. And I guess particularly the students appreciate to see the advantages in the last years and possibilities. And after giving a number of nice examples, I personally also appreciate you mentioned John Spence. He was a tremendous character, and he was with Henry Chapman and others launching serial crystallography, which is now also a method at synchrotron radiations. And he was a tremendous character. We met several times, and the community certainly will miss him. And I hope we can collaborate also together with our colleagues in Karachi on compound screening in future on COVID, if we can merge this enterprise. And I guess now the session is open for some discussion and questions. I'm not sure how it is arranged, if the participants can comment in chat or ask direct.
Professor Christian, thank you very much. Yes, we are open for question and answers. Professor Samar, thank you very much for a wonderful lecture. And if you allow me, can I take some questions for you?
Yes, please.
So first question is from Ms. Maria Khalid. She's asking what types of bioinformatic analysis can be done to evaluate structural changes in a protein due to mutation.
Well, that's a very good question. So it depends on the prior knowledge one has from the structure itself, and then using that, there are many methods which you can begin to use. So in bioinformatics, you can combine molecular dynamics as well, and then you can begin to look at it. But many of these structures and the mutations are unpredictable. Our knowledge base is increased, even though we have 150,000 structures. It is still a small subset of the protein family, and mutations can be unpredictable.
Right. So we have another question from Dr. Najibullah. He's asking how would one compare single particle tracking analysis, that is like having many states of the protein with ligand in time scale, with a serial crystallography?
So you need to get your biochemistry of the system very well defined, so that when you mix, say in the case of mix and inject, then each crystal is soaked through the mixing chamber for the same amount, and then you can look at each crystal shot providing it's like a stop flow system. The size of the crystals are very small, and that's the beauty of the SFX mix and inject system, because SFX can be applied on very, very small crystals, and therefore uniformity of mixing is very, very, very high compared to a normal crystallography where the crystal size is large. In the case of the pump and probe approach, then again that approach is designed such that because the crystals are thin and small, then by defining your chemical the chemistry using other methods first and then using it for SFX is highly successful. So there are several examples which have now been done for both mix and inject system as well as the pump and probe, which show that SFX approach is very good. SFX is the only approach you can use in that sense. SF-ROX will not be the right approach to use.
Thank you, sir. So here is another question from Dr. Wasim. He's asking, what is the best way to approach building a custom polypeptide?
Can you repeat the question again?
Yes. He's asking, what is the best way to approach building a custom polypeptide?
All right. I guess so. This touches upon a new and emerging field where one can talk about synthetic biology, where from using the structures and the templates of the catalytic core, you can begin to have a designer enzyme or designer peptide for that purpose. But of course, peptides are also used for as candidate drug molecules. So that is again another approach, but it will depend on the knowledge base you have and how responsive and how stable the structure will be responding to the peptide.
So here is the last question from Dr. Wasim again. Is there any freely available algorithm or a mathematical model that can predict aggregability of a protein?
Well, there's been a lot of success, and because our knowledge base is increased and the structures are increased, then use of artificial intelligence is being made extensively, and there's been a major success of AlphaFold, and which was hit the news only about a few months ago, is a big success. So there's a significant amount of work which is going on, which uses the knowledge from the experimental data into being able to predict how the proteins might aggregate.
Thank you very much, sir. With this, we come to end of the session. I would like to thank you and Professor Christian for your valuable time, and it was wonderful having you both here. And I would also like to thank our participants for giving their time and joining this important lecture. Thank you very much, and hope to see you all in another exciting lecture of Comstech. Bye-bye.
Thank you.
so much.
Bye-bye.
Thanks very much. Bye-bye.
Bye-bye.